Thank you so much, Steve. A tremendous pleasure to be here. In fact, I've already been having so much fun talking to people that I'm hoping I have enough catecholamines left for today's talk, because we know, or at least you will know by the end of the talk, that prefrontal needs that, and prefrontal needed for this talk. So especially, we have so much the same goals, but from a complementary perspective. So trying to understand how genetic insults affect signaling pathways that then affect this circuit function and lead to symptoms. And we're trying to bridge that by really trying to see how these signaling pathways interact to specifically affect circuits in layer 3 of dorsolateral prefrontal cortex, which are so tremendously affected in mental disorders like schizophrenia. And what I'm going to be telling you today is we are finding that these layer 3 circuits are regulated at the molecular level in a way that's fundamentally different from classic circuits. For example, fundamentally different from the primary visual cortex. And we've just started studies, as I'll show you some of the data, where we're actually recording and looking at V1 as a direct comparison. And our overarching hypothesis is that these unique regulatory events are part of what makes dorsolateral prefrontal cortex, and these circuits in particular, so vulnerable in cognitive disorders such as schizophrenia, and also, I think, actually in age-related disorders like Alzheimer's. And already, if you look at the architecture of these cells, you see how special they are. So if you compare, this is now rhesus monkey, layer 3 dorsolateral prefrontal to layer 3 V1, more than twice as many spines, twice as many connections. And if you look over primate evolution, from simple like Galago all the way up to human, what you find is the number of spines is really the same in V1, whether or not you're human, Galago, whatever. But in dorsolateral prefrontal cortex, you see this vast expansion on the number of spines from early primates. Here's rhesus monkey, and here's human. So this is where we're expanding. It fits with the vast increase in the number of connections needed for higher cognition. And we see this in the basal dendritic tree as well, where it becomes quite simple, rhesus monkey more complex, and most complex of all in human. So we're looking at the circuits that are evolving most in primate evolution, and that are also very different from rodents. So this is human dorsolateral prefrontal cortex, and this is mouse and rat medial prefrontal, since they don't have a dorsolateral region. And one of the things that I think is really important for the entire field to remember is in humans and rhesus monkeys, layer 3 is immense. And these cognitive circuits that we focus on, that Pat Goldman-Rakesh discovered, and that are so affected in schizophrenia, particularly in deep layer 3. Whereas you see layer 3 is very tiny in rodent medial prefrontal. Conversely, layer 5, quite small in human and monkey, and very large in rodent. So some of the differences then that we might be seeing and thinking of as species differences might be laminar differences instead, and I'm hoping there can be more research on layer 3 in rodents to see if there's things that might be more similar to human and might be a hopeful way forward. So when we talk about differences, to keep this in mind, because so much of the physiology in particular in rodents has been done from layer 5. Layer 3, I say, is particularly relevant to these disorders, particularly deep layer 3. And this is where we see the loss of neuropil and the loss of dendrites in spines in schizophrenia, this now iconic image from David Lewis's lab. And David has also now shown that these deep layer 3 pyramidal cells are profoundly hypometabolic, that they are likely the primary deficit in schizophrenia, whereas the GABAergic surrounding interneurons, a compensatory weakening to make up for this loss of connectivity and firing. And we can see this reflected at the imaging level, where hypofrontality of dorsolateral prefrontal correlates with symptoms of thought disorder. So this is not just an epiphenomenon, it is likely deeply related to the distortion in cognitive thinking. I wanted to add that these circuits are also really important for age-related disorder. These layer 3 pyramidal cells are what fill with neurofibrillary tangles and die in Alzheimer's. And this is what's correlating with the onslaught of cognitive impairment. I don't have time for that in today's talk. I just wanted to compare. In comparison, area 17, primary visual cortex, almost no tangles, even in end-stage Alzheimer's disease. So I think this is a fascinating lesson, what makes these guys so resilient, these guys so vulnerable. We're beginning to see how phospho-tau accumulates in these cells and makes them vulnerable. And if you're interested, we had a paper in PNAS last year, and we're continuing to build on that. And just to say that I think the aging rhesus monkey is a really excellent model for asking these questions of why some cells are vulnerable with age and others not. But I'm being disciplined and not talking about that today. And instead, so we can focus on schizophrenia and questions more immediately related to Stanley. And so what is making these layer 3 circuits so vulnerable? I think part of it is this differential regulation. And so let's first review what classic calcium cyclic AMP signaling does, for example, in hippocampus, in a dish, in developing visual cortex. So the traditional role, here we have glutamate release and NMDA, NR2A. We have calcium coming in. This is our spine apparatus, which is the extension of, let me just fill in here, NMDA and Ampa receptors. So Ampa stimulation by glutamate allows NMDA to open. Now we have the calcium entry, which can, in many spines, cause more calcium release from the spine apparatus, which is the extension of the smooth endoplasmic reticulum into the spine. And if we get enough calcium activation of cyclic AMP, protein kinase A signaling, so we have feed-forward signaling here as pKa phosphorylates IP3 receptors, causes more calcium release. And if we have enough of this from a very strong synaptic connection, we phosphorylate Krebs and get transcription, where we now create a strong, mature spine. And in these classic synapses, NR2B, NMDA are often extrasynaptic, whereas the A subunit ones in the synapse itself needing Ampa receptors to be able to sit in a depolarized membrane and open. So that's classic LTP plasticity. And phosphodiesterases, PDE4s, chew up the cyclic AMP and stop it and weaken plasticity. Oops. And evidence of this in hippocampus NV1. The other common place for cyclic AMP signaling is presynaptic, where cyclic AMP PKA activation primes vesicles and increases transmitter release, often glutamate release. So these are the traditional roles of calcium cyclic AMP signaling. And what we're finding is that these signaling pathways do the exact opposite in dorsolateral prefrontal layer 3. Instead of strengthening, they weaken connections and do so by opening a variety of ion channels, KCNQ, Hcn, that are themselves and have increased opening by cyclic AMP or pKa signaling. And so that is a lot of the data I'll be telling you about today. And this idea that we have these built-in mechanisms to weaken a connection. If it's not regulated properly, you have a genie out of the bottle who can weaken and destroy connectivity. So we are currently comparing dorsolateral prefrontal to V1 in the rhesus monkey to directly compare how modulation works. And again, V1 little affected in Alzheimer's, modestly in schizophrenia, where marked effects in dorsolateral prefrontal. And our approach is threefold. We do immunoelectron microscopy to actually see where proteins are and who they're interacting with in these circuits. We do physiology, where we're recording from these neurons in monkeys performing relevant cognitive tasks. And we ionophorese, which means electrically sprinkle just a tiny amount of drug onto the cells we're recording from to see how it changes their physiological signature. And this is such a small amount of drug that it doesn't alter behavior, but it gives us a good idea of how these subtle perturbations alter a neuron's response. And then the third thing we do is behavioral studies, where we give drugs systemically in the monkey to look for things that might be potential cognitive enhancers. We also do some studies where we infuse drug into rat prefrontal to look at effect on working memory, and occasionally local injections into monkey prefrontal as well. So this threefold approach to try and understand how these circuits are modulated. I'm going to start with V1. We have the least amount of data, and these studies are still ongoing. And what we do is the monkeys are fixating and looking for this fixation spot to dim a little, and they press a button when they see a dimming. But as they're doing it, we're flashing these fields, identifying the receptive field of the neurons in V1 that we're recording from. And these are classic feed-forward sensory circuits. And when we've been looking now at what does cyclic AMP calcium do in these circuits, and when we look at the EM level, we see evidence for this kind of classic signaling with a mushroom spine, where PD4A is localized in these spines. So probably in the past, when plastic changes occurred, that this was indeed involved. But mushroom spines are actually just a minority, a small minority of the spines in V1. And they are a minority of where we see cyclic AMP signaling proteins. The main place that we see them is in these glutamate axon terminals. And if we look blown up here, we see them concentrated around the vesicles near the release zone. And so we think that what's going on is the classic pKa priming of vesicles to enhance release. And indeed, when we do recordings from monkeys and ionophorese cyclic AMP agents, the data are fitting with that. So here is a neuron under control conditions in black responding to a visual stimulus. And now when we give increasing doses of 8-bromocyclic AMP, which mimics cyclic AMP and activates pKa, we see a dose-related increase, large increase here. Also background firing, but this is greater than this, so it's an enhancement of sensory firing. So this fits with this working model, and it's something that's been seen throughout the nervous system for years, that cyclic AMP enhancing the firing. And although I'm not presenting the data here because it's still being recorded as I speak, I should say that we see dramatic effects of Ampa receptors, so that if you block Ampa receptors in these monkeys, tremendous loss of firing. Also loss of firing if you block NMDA. And I wanted tell you that because you'll see it's very different once we get to prefrontal. So in quick summary of our preliminary data from V1, we're finding that cyclic EMP-related signaling strengthens the visual sensory processing of these cells, and that these are needing Ampa and NMDA actions. So no surprises, but we needed to validate that this was indeed true in the primate V1. And now what we're finding in prefrontal is totally different. And the role of prefrontal is totally different. What it is trying to do is represent information, for example, visual space, in the absence of sensory stimulation. And this is the foundation of abstract thought. We've been testing it, as you'll see, in working memory paradigm, but also essential for top-down executive control. So this is very much the work of Patricia Goldman-Rakish. And if anyone is interested, I did this review on the 10th anniversary of her death, so it came out in 2013, trying to capture the highlights of the many things she did to truly transform this entire field. Because what she gave us was the microcircuitry, the neurobiology of visual spatial representation. So in her work, and we continue this, the monkeys are doing a visual spatial working memory task where they're again fixating on a central spot, and as they do so, a cue comes on in one of 8 locations for just a half a second, goes off, and now the monkey's having to remember that location over a delay period of several seconds. And then the fixation point goes off, and that's the signal for the monkey is now able to respond and move its eyes, or in some paradigms, move its hand. And if they get the correct location, they get drops of their favorite juice reward. And over a typical daily session, there are hundreds of trials where the cue randomly changes its position. So the contents of working memory are constantly having to be updated. And this absolutely requires dorsolateral prefrontal cortex. I keep, here, I'm going to do this so I don't keep hitting that. So what Pat found, and these were studies with Charlie Bruce and Shintaro Funahashi, is that there are what she called delay cells in dorsolateral prefrontal that represent visual space. So here is a neuron, for example, that fires across the delay period on every trial where the cue had been here, but not any of the trials where the cue was in these other locations. So here's fixation, the cue period, delay, response. All these trials, this neuron fires if the cue had been here and it's remembering that location, but none of these others. So you can see if you get to the end of the delay period and this neuron is firing, the brain knows it was here, look here. Whereas if it's not firing, it knows it wasn't there. Does that make sense to everybody? So this is the neural representation of visual space. And that in itself was an enormous advance. Pat also found more typical sensory and motor cells in dorsolateral prefrontal, some cue cells that fire only when the cue is on, response cells that fire just before the motor response and are probably conveying this information to the motor system. And very interesting, these cells we call post-saccadic response cells that fire during or just after the response that appear to be the corollary discharge, the feedback from the motor system saying, this is what I have done. These in particular are really intriguing. They're altered by dopamine D2. I'm not talking about them today, but I think they're really important for schizophrenia as well, where there's evidence of weakened or altered corollary discharge, but not for today. But I did want to make the point that these cue and response cells have a totally different pharmacology than our delay cells. They're huge Ampa response. You block Ampa, they stop firing. And we're now seeing some evidence that these are also enhanced by cyclic AMP, much more like the V1 cells, much more what's like has been seen in rodent. And again, is this because rodent studies are done in layer 5 or because there's species differences? We don't know. But I just wanted to put that on everybody's radar screen, that you can't just say prefrontal. Layer is really going to matter. Circuit is really going to matter. And let me now talk about this unique regulation and the circuitry for these delay cells. So Pat discovered these microcircuits in deep layer 3 that are comprised of pyramidal cells that excite each other, recurrent excitation. So they're all interconnected in these long horizontal connections. And this is what mediates this persistent firing and allows the cells to fire even without sensory input because they excite each other. And I'll show you that this is utterly dependent on NMDA receptors. And then the cells also have this beautiful spatial specificity. If they were firing to every direction, there'd be no information. Some of this is due to the inputs from parietal association cortex being somewhat specified, but it is enhanced and refined by lateral inhibition from GABAergic cells. So for example, if the cue's at 270 degrees, this group of 270-degree pyramidal cells are firing. They activate a basket cell, which is a GABAergic interneuron, to shut off our 90-degree cell so it is not firing under these conditions. So lateral inhibition refining the spatial tuning and allowing us to have precise information held in working memory. Now, these are the very circuits most vulnerable in schizophrenia. Pat and Lynn Selman were the first to show this very reduced neuropill, specifically in deep layer 3. And then David's lab going on to show the loss of dendrites and spines that's most accentuated in deep layer 3. And this is probably due to a combination of errors in circuit formation in utero and then accelerated loss in adolescence, which I wonder how much of this is stress-driven as well, given the links between stress exposure and onset of the disease in late adolescence. And then there is also this weakening of the GABA that David has shown, compensatory weakening of the interneurons, but he thinks now a primary deficit in these pyramidal cells. So we would then have weaker persistent firing and noisier signals. So being able, not being able to hold well-defined information in mind and thus contributing to thought disorder. And I've wondered how much this relates to these waves of gray matter loss in adolescence. So I'm hoping that our work might be able to give some insights onto that by studying the needs of these circuits and why they're so vulnerable. So what we're finding is that the arousal modulators have essential actions on whether these connections, these recurrent excitatory connections, are actually effective, and that this is what allows mental flexibility to coordinate with our cognitive and arousal states. So why you're all, even though it's 3 in the afternoon, so alert and interested, because the data is so fascinating, and because you all had coffee, which then increased acetylcholine. And then what these guys are doing, what we're finding, is they can very rapidly and reversibly alter the strength of these NMDA synapses, either by opening potassium channels to rapidly weaken a connection, or if we're interested, as you all are now, and feel safe, inhibit the cyclic AMP signaling, close these channels, and strengthen these connections. And as I said, this idea of this built-in mechanism to weaken a connection confers vulnerability when not properly regulated by genetic or environmental insults. So now let me show you some of the data. First of all, that NMDA receptors are there in the synapse. And one of the first things that we see that's different is NR2B, which are extrasynaptic in your classic synapses, are completely in the synapse, only in the synapse, in these layer 3 circuits. So the only place we see them is in the postsynaptic density, none extrasynaptic. And when we block NMDA receptors, we see this complete loss of representation. So here's a neuron representing 90 degrees. Now when we block, including if we just block NMDA receptors with NR2B subunits, can you see how that representation's gone now? So there's no longer any information held in mind. We get the same thing if we block NR2A or if we block both of them. So they're all important. But what's interesting is that NR2B is as well, because these guys close slowly. They allow a lot of calcium in, and that's probably part of the vulnerability profile. So yes, and I just told you the A as well, so both are important. And in contrast, Ampa, not so important. So here's firing for the neurons. Just so this is the preferred direction, and this is one of the non-preferred directions. So I'll mostly just show you two to save space. So here's a neuron in blue firing for its preferred direction under control conditions, doesn't fire for non-preferred. If we block NMDA with the NR2B, you can see this dramatic loss of firing. But if we block Ampa, very subtle effects here in green. The only time it's significantly different, I'm sorry the standard error is hard to see, but it's quite wide. This is the only segment where it's at all statistically significant, and even then it's not much. So we were really surprised by this because, hey, AMPAs are essential for NMDA. This is impossible. So what's relieving the magnesium block that allows NMDA to open? And the answer turns out to be acetylcholine is playing this key role. And our published work is showing acetylcholine is doing this through nicotinic alpha-7 receptors. And they're located right in the glutamate synapses where the NR2B are. So here's a glutamate axon, and the EM is showing the alpha-7 are right there. They're also nearby in this cholinergic synapse. And you can see these spines are really thin. They're long and thin. And we think that architecture is essential for allowing these gaining actions to allow these ion channels to have such a huge effect. So you can depolarize this tip and allow NMDA to open. And let me show you some of these data now, that when we block alpha-7, we get a beautiful loss of firing, and conversely, when we stimulate, we get a beautiful increase. So here's a neuron firing under control conditions and then with increasing doses of an alpha-7 agonist. And this is important for NMDA. We can rescue firing after blocking lightly NMDA receptors. But this is the experiment that I found most interesting. Normally, if you spritz some NMDA on these cells, you get increased firing. That's not surprising. But if we first block alpha-7, the NMDA is no longer able to excite the cell. So it suggests you have to have these guys depolarizing the membrane for the NMDA to have an effect. And we're seeing similar effects now with acetylcholine stimulating muscarinic M1. We don't know if they're in the same synapse, but it seems like acetylcholine is playing this really key role in these prefrontal circuits. And what's fascinating to me is acetylcholine is only released when we're awake or in REM sleep. So not in deep sleep when we're unconscious. So is this part of the basis for conscious thought? That having acetylcholine, allowing these networks to connect, is allowing us to be conscious. So I think that's a really fun thought. It's also very relevant to schizophrenia, where there have been long-term genetic links to weaker NMDA and alpha-7. And is this why so many patients with schizophrenia smoke? To strengthen these weaker connections. And I have heard patients describe themselves when they're smoking that they feel like they are appearing to themselves. And is that because they are bringing more of these circuits online? And based on this long history and this understanding about smoking, alpha-7 agonists are now being developed as potential cognitive enhancers for schizophrenia, ADHD, age-related disorders. And our data suggests that this could be a really fruitful strategy, but you really have to watch out for dose. So here's a neuron under control conditions, weakly representing 270 degrees. With a low dose of alpha-7, do you see how that information is beautifully accentuated and how helpful that would be? But if you go too high, and you can go too high really quickly, now we have non-specific excitation. And the information contained is actually lost because now the cell's just firing to everything. So dosage will be critical in developing these agents as cognitive enhancers. Now I want to turn to the next really important phase of this, which is calcium and cyclic AMP signaling. Now we've gotten our NMDA receptors open, we've got calcium coming in, and we're discovering this whole extraordinary apparatus which provides negative feedback, allows network gating, and allows us to disconnect and weaken these connections very rapidly. So we see that most of these synapses in layer 3 have a well-developed spine apparatus connected to the smooth ER with these calcium cyclic AMP signaling, evidence of this cartoon right there, and I'll also show you with these nearby ion channels. So let's show you the EM to support the cartoon. First of all, we have evidence that ACAP6, anchoring protein 6, anchors pKa to the spine apparatus. So here's the dendrite, here's the spine neck, the spine head, the synapse, and the glutamate axon terminal. And in pink here, you can see the spine apparatus extends into the spine and elaborates in the head. And all along it, we have ACAP6, these gold particles, which are anchoring pKa to the spine apparatus. And there's also data, not from my lab, from others, showing that mGluR 1 and 5 are both localized here, positioned to drive that. We also think norepinephrine alpha-1 receptors might be there, but we've yet to do those studies. So these would be through IP3 generation driving this, so they're positioned there. And we do have some physiological data to support the mGluR 1 and 5, but I don't have time to show that to you now. Instead, I want to go on and show you how these ion channels are localized there. First of all, Hcn channels, which open with hyperpolarization, but their open state is also greatly enhanced by cyclic AMP. And we find that these Hcn channels are tremendously concentrated in these spines right next to the synapse and in what Konstantinos calls the thin bottleneck, the points in the spine neck where it gets especially thin. The channels can ring it in a way like nothing's going to get through. And when we block these channels with a low dose of ZD7288, we get enhanced firing and specifically the preferred direction. When we go to high doses, we get a collapse in firing, probably because now we're blocking the Hcn channels on the distal dendrite that normally serve to depolarize the entire apical dendritic tree. But this was what was most interesting to us, that we could get this enhancement as if we're allowing in more of this network. We're also seeing a whole lot of Hcn channels, 2, 3, and 5. And Hcn channels are opened by pKa signaling, specifically Hcn-Q2, or if they're combined with 3. And because of the work being done at the Stanley, I wanted to mention that there's work showing that GSK3 beta might interact with pKa in increasing this open state, which may be particularly relevant to bipolar. We have not done work yet to see if that's there, but I wanted to put that out there to try and link it to some of the work being done here. So the EM is showing all 3, 2, 3, and 5. We can find all of them on spines in layer 3. And we get very dramatic effects when we block them. We get the enhanced firing. And when we open them, we get the reduced firing, as we would expect. And then we can drive this by activating pKa. So here's firing under controlled conditions. And now if we give the 8-bromo, we get a reduction in firing, which is exactly opposite of what we saw in V1. And we can reverse this by blocking Hcn channels. And I'll show you in a moment, we get even greater effects when we block PD4 and allow cyclic AMP levels to build up. So we have both of these channels. And now let me show you how dopamine through D1 receptors can drive this through increasing cyclic AMP. Dopamine D1 have complex effects, so if you'll stick with me, I'll show you this. We have these preferred network inputs, where here a 90-degree cell is getting a 90-degree input. Also non-preferred network inputs, so here's 120 degrees. And I'm going to show you that D1 can help carve away this non-preferred when we have the dose just right. So the EM is showing location of D1 receptors, some of it right in the synapse, in the postsynaptic density here. And so I've put that there. And the evidence suggests that, this is now from rodent, but it fits with some monkey data, that D1 right in the synapse through pKa is probably phosphorylating NMDA receptors and keeping them in the synapse. And if you give huge doses of D1 antagonists, you do indeed stop all firing. The other place we're seeing them is right next to the Hcn channels. And I've drawn these as the cartoon out here. They're usually right next door. And what we see when we activate these is first sculpting actions, where we take this noisy firing from the non-preferred and sculpt it away. And you can see that this creates a much better refined representation. So here's the neuron firing under control conditions for its preferred versus non-preferred directions. When you give D1, you really refine this. So now you have sharper tuning and just firing for the preferred. But when we give higher levels, which likely occur during stress exposure, now we have this huge loss of firing, and we are reducing the preferred network inputs as well. We also see this huge loss of firing when we give norepinephrine alpha-1 agonists. And this is through protein kinase C, in that if we inhibit PKC, we can restore firing. And this ends up being especially important with chronic stress, where in rats now, chronic stress leads to loss of spines from layer 3 neurons. And we have this correlation with loss of working memory. But if we inhibit protein kinase C every day before the stressor, we can rescue spines and rescue working memory. And we've wondered if in part this has to do with PKC phosphorylation of Marx, which disrupts the actin cytoskeleton. So these acute effects of stress, we think, contribute to the chronic stress profile as well. Well, thankfully, there's a whole bunch of endogenous mechanisms for regulating this feed-forward signaling and allowing the networks to connect. And I'm just going to show you a couple of these because they end up being so relevant to schizophrenia. So PD4A, phosphodiesterase, turns out to be the phosphodiesterase that is here next to the spine apparatus. So in pink, we see the spine apparatus. This is ACAP6, anchoring PKA, and the green arrows indicating the PD4A throughout there. And what's anchoring it in the correct place is DISC1, disrupted in schizophrenia. So here's our glutamate axon terminal, our synapse, the spine apparatus, which continues there as well. This large gold particle is attached to an antibody that recognizes DISC1. These three tiny gold particles are attached to antibodies that recognize PD4A. And you can see this is less than a nanometer. So DISC1 anchoring PD4A to the correct location to keep all of this under control. And indeed, if we inhibit PD4, we see this total loss of firing that we can rescue by blocking these channels. So physiological evidence for this interaction as well. Interestingly, inflammation impairs the ability of DISC1 to anchor PD4A5 specifically, not the other phosphodiesterases, just this one. So it's a way where we could, and this is this paper if you're interested in it, a way that inflammation can lead to dysregulation of this pathways and weakening of this connection. I wanted to share with you some of our newest data that's not yet published, but it's directly relevant to schizophrenia, and that's mGluR3. mGluR3 is traditionally thought of as localized only in glia, in the perisynaptic astrocytes. And what we're finding is it is indeed in glia, but the other major place it is in layer 3 of dorsolateral prefrontal is these exact thin spines. Postsynaptically, there it is right next to the synapse, right next to the spine apparatus, positioned to inhibit cyclic AMP signaling. And indeed, when we stimulate these receptors, we're seeing enhanced firing. And we're working with Jeff Kahn's group at Vanderbilt, hoping they can create a selective mGluR3 PAM. Because all we've been able to use so far are these mGluR2/3 compounds. And mGluR2 are localized in particular presynaptically. There are some post and in glia. But the presynaptic actions eat away at and diminish the efficacy of these agents. And we think this is why they might have failed so far in treating schizophrenia. So in schizophrenia, then, we're seeing this whole constellation of things that normally strengthen these synapses being decreased or weaker in schizophrenia. The one thing I didn't talk about is RGS4, which is positioned to inhibit GQ signaling right there. So these things that strengthen are all weaker. And then conversely, the things that weaken the synapse are increased in schizophrenia, mGluR1 and D1 early in the illness. And I've also wondered, this is a speculation now, but again, based on the, sorry how that keeps happening. Based on the GWAS data, whether the L-type calcium channel 1.2 is often focused in spines. It is increased actions by protein kinase A. It's known that the variant in schizophrenia increases its activity. And it would then, through stimulation of, for example, ryanodine receptors, drive this weakening process. So I think this would be a wonderful target for us to look at next to see, is it indeed in this position and having these weakening actions. Now, it also shows how we can have this whole variety of genetic insults that would lead to the same phenotype of weakened connection in the circuit most needed for higher cognition. And now I want to close on a potential therapeutic that's already successfully translated to humans and that I'm hoping might be useful in schizophrenia as well. And that's the noradrenergic alpha-2A receptor, which is also here on these spines positioned to inhibit cyclic AMP signaling. So the EM shows it right there next to the Hcn channels. Sometimes it's right next to the synapse, and sometimes it's in the spine neck. And if we block these receptors, total loss of firing that we can rescue by blocking these channels. So these data really made me a believer. And if we infuse large amounts of yohimbine into dorsolateral prefrontal cortex, huge loss of working memory, increased impulsivity, impaired regulation of behavior. This is the work from Baoming Li's group in China, who's been a long-time collaborator. Conversely, if we stimulate this receptor with guanfacine, which is currently the most selective alpha-2A agonist available, we see a beautiful enhancement of firing. And when we give it systemically, a beautiful improvement in working memory, reduced distractibility, and enhanced impulse control. And we have been able to see with systemic guanfacine improvement in working memory in mice, wild-type mice, but not in alpha-2A mutant mice. It's a small effect. I think if we didn't know what we were looking for, we wouldn't have found it. But it gives me hope that there is this small signal there in the mouse. And when we give the chronic stress, where we get the spine loss, if the animals are treated daily with guanfacine prior to the stress, we rescue the spines and we rescue working memory. So it protects gray matter as well. And some of this may be due to guanfacine's anti-inflammatory roles. Activated microglia express alpha-2A receptors, and guanfacine deactivates these microglia and has anti-inflammatory effects. So both of these actions might be very helpful in protecting prefrontal cortex. So based on our work, guanfacine is now in widespread use for treating prefrontal disorders in humans. It was approved by the FDA in 2009 for the treatment of ADHD under the brand name Intuniv, which is extended release formulation for children because they metabolize it so quickly. It's used off-label as a first-line treatment for Tourette syndrome because strengthening prefrontal allows subjects to inhibit their tics. It's used off-label in autism spectrum disorders to treat the disinhibition, the ADHD-like symptoms. And an area where it's particularly useful because not much else is in treating emotional trauma, PTSD in kids, which often evidences as oppositional or aggressive symptoms, especially in boys who've been traumatized. And it's one of the few things that helps these kids to control themselves and calm themselves down. It's also being tested in a number of other prefrontal disorders where it's still mostly laboratory, although it's extending into the clinic as well. So Tom McAllister is finding it useful in treating mild traumatic brain injury, which so often affects prefrontal. If it's extensive prefrontal deficits or destruction, obviously there's no substrate left, but it can be helpful in milder. The Yale Stress Group is finding it really helpful with substance abuse, finding it helps people quit smoking, and helping people who are cocaine addicts to resist. In particular, helping them when they're stressed to have the self-control to not pursue it. An anesthesiologist at Vanderbilt uses it to treat emergence delirium from anesthesia. He describes it, he thinks what's happening is the prefrontal is still disconnected, so when he's trying to talk to them to tell them it's okay, you know, this big thing in your throat, don't worry, we'll take it out soon. And they can't understand him and panic, and it can be life-threatening because they try to take out the breathing tube. So he now has developed an IV form of guanfacine, so he can give it right away. And the group in London is finding it useful for treating strokes and infections that generally afflict the association cortex. And part of my visit here, really hoping it could help prodromal schizophrenia, perhaps help to slow or even prevent that wave of gray matter loss from prefrontal at the onset of illness. So I'd so hope that we can collaborate and that help with understanding and treating schizophrenia. And I'd like to close with many thank yous. In particular, the physiology was done under the supervision of Min Wang, the EM Konstantinos Paspalis, and thanks to all of you.